But the most widespread technique for understanding brain functions is to record the brain's electrical activity. Because neurons are biochemical electrical generators, neuroscientists have found ways to record the nerve signals coming from a single neuron as it responds to some event. The electrical activity of the entire brain can also be recorded. This brainwave pattern is known as an electroencephalogram, or EEG. It represents the total activity of the neurons in the cortex. E. Roy John, director of the Brain Research Laboratories in New York, is a leading proponent of the new science of neurometrics, the precise electrophysiological measurement of neural functioning.
We and scientists in other laboratories have looked at the electrical activity of the human brain across the age range from early childhood to old age, specifically from 6 to 90, and described the electrical activity quantitatively by analyzing it using computers. What we have found is that this electrical activity changes in a very orderly way, which can be described by simple equations. The basis of neurometrics is that every individual's brain electrical activity is compared to these normative data which we have collected across the lifespan. The way this comparison is made is statistical. The data are presented in a way which is intended to attract your attention to abnormal features because there's such an enormous amount of information that's being evaluated by the computer. So the information is color-coded. If brain electrical activity in a particular anatomical region is normal, that part of the image which the computer presents is an earth color. To the extent that there is an excess of some kind of activity in a particular region, that region becomes colored red. The more abnormal the excess, the brighter the color of red, going up to orange. If there is something lacking in an anatomical region, if it's deficient in a certain kind of activity, that region becomes colored blue. When abnormalities are present, they appear as blotches of color in certain regions. We have now patterns that identify the major psychiatric disorders, depression, dementia, schizophrenia. We can recognize alcoholism. Neurometric analysis is a functional analysis. It reveals abnormal transactions between brain regions. Those regions may interact abnormally for several reasons. One is because their anatomical structure is abnormal, which we can see. Two is because in spite of normal structure, their neurochemical reactions are abnormal, which we also see. Even with normal neurochemical capability and normal anatomical structure, the brain can enter certain states which are abnormal. For example, we had a patient come in for an examination who was a staff member here at the university, who was known to the technician who was performing the examination. Really wasn't a patient. The person came in as a member of our normal database. An examination was performed and the results were perfectly normal. After completion of the examination, the technician who knew this volunteer asked her a personal question about her boyfriend. And in fact, this person had had a very distressing rupture of her relationship with the boyfriend. While she reacted to the question, accidentally the recording continued. And that recording was analyzed and showed pathological depression. So here we had, in a matter of a couple of minutes, a brain move from a perfectly normal picture to a pathologically depressed picture, and in fact, a couple of minutes later back to a perfectly normal picture. That was not abnormal neurochemical capability or structure. That was a state. And what we can see is the change of states as people think of different things, as they have different moods. Almost everybody knows somebody who's been in countless years of psychotherapy with very little apparent benefit. Many of those treatment-resistant patients, in fact, have pathophysiological causes for their behavioral distress, which can now be recognized. Psychology for a long time has dealt with the organism like a black box. Something comes into it and something comes out from it. And you infer what's in between by the relationship between the input and the output. With those inferences, bright people can learn a lot about what's in between, but it will never compare with being able to open the box and look inside, and that's where we're at now.
While some neuroscientists explore the electrical activity of the brain, others are studying its chemical activity. One of the most promising avenues of research today is the effect of drugs on specific functions of the brain. The brain itself is a biochemical drug factory that manufactures opiate-like molecules. These molecules are called opioid peptides, part of the complex system of neurotransmitters that send signals from cell to cell and ultimately to other organs of the body. Although there are many different types of opioid peptides, they are known collectively as endorphins. These endorphins also come with corresponding opioid receptors throughout the body. There is a finely tuned division of labor among them, as each endorphin follows a specific path within the nervous system. The action of each one is mediated through different types of receptors, and what all of them do for us is nothing short of remarkable. Endorphins, like narcotics, can create the mood of euphoria that runners experience and reduce pain, depending on which peptides attach themselves to which receptors. In fact, they play a major role in most experiences that involve pleasure or pain. Research has implicated them in a host of our emotional reactions, from laughing and crying to physical arousal such as fighting. And endorphins are but one of a staggering number of chemical influences on the brain. At the University of California at Berkeley, psychologist, neuroscientist Joseph Martinez is investigating how brain chemicals also affect learning and memory.
We find certain chemicals in the brain that we call neurotransmitters. And we think that some neurotransmitters are important for learning in animals and humans. And by knowing what these chemicals are, we are able to mimic how the brain talks to itself. And we can coax the brain into either forgetting an experience, or we can make the brain actually remember better by stimulating some of these chemical systems. In our experiments, we use very simple learning situations. We have a maze which has two arms, and we put the food into one of the arms, and the mouse runs down and obtains the food. The mouse learns very quickly where the food is, and he always runs to that particular arm. Even though we've trained them in this maze task, their memories are not yet fixed. They're not yet permanent. They're not yet into long-term memory, because the brain is still processing this information. And since it takes a period of time to become permanent, we can interfere with this process by injecting the animals with drugs. In this particular experiment, one group of animals we had injected with a drug called scopolamine, which blocked the transmission of information in the brain. And the other group of animals had simply received a saline injection. The animals are trained in a drug-free state. They don't receive drugs while they're learning the maze.